Once upon a time, you were bored, then suddenly you thought of ants. So you took a pinch of sugar and placed it on the table. After a few minutes, you saw one, then two ants on the table. They might be just wandering aimlessly in your house, or they might be actually looking for food. And after a few bumps here and there, they were able to find the sugar on the table. This is kind of similar how phagocytes work. Here is the overview of phagocytosis. There are many steps to follow. I think there are actually more in the more detailed one, but this is what is provided for us in the syllabus, so we will stick to this one. So for phagocytosis, we will be talking about this one step by step, starting with this one first. First step, you have attraction or chemotaxis. In this step, resting phagocytes may accidentally bump into a pathogen, just like the ant earlier. Or B, the phagocytes sometimes move towards a chemical attractant. And in the case of the ant earlier, it is the smell of the sugar. Okay? And for phagocytes, chemical attractants can include that can signal the presence of a pathogen. And once they do detect these chemical attractants, they gain the ability to leave the capillaries and enter the tissues toward the site of infection. This process is known as chemotaxis. When they move towards the site of infection, phagocytes switch to a higher energy level as it chases the pathogen. When I say chase, I actually mean that the phagocyte is undergoing directional movement towards where the chemical attractant is. This now leads us to the second step, recognition and attachment. Remember that different cell types express different receptors. But some receptors are general, meaning they can identify a self-produced molecule versus a potential threat. And others are very specific. The pathogen will not initiate phagocytosis without successful binding of the cell surface receptors. In this case, you could see the receptor in here floating around, and this one will be bringing it to the phagocyte just like this one. This one in here might not move out, but it might just, wait for a bacteria or a pathogen maybe to attach to it. Number 3, endocytosis. In endocytosis, the phagocyte uses extensions of the cytoplasm known as pseudopods. Pseudo meaning false, and pods mean feet. Okay? So in here, the pseudopods will protrude toward or outward either side of the pathogen until both sides meet and the virus or the pathogen is enclosed, creating a pocket-like indentation that we call as phagosome. Looking at this image that we have in here, we have the edge of the plasma membrane that is opening up, right? This edge will be increasing until they meet and close like this. And once they close like this, it means that this pathogen will now be enclosed in a membrane that we call as phagosome. The phagosome is where the plasma membrane has moved around the particle or the pathogen, encasing it safely inside the cell. which leads us now to the next two steps. So step number 4 will be fusion of lysosome and phagocytic vacuole, and step 5 will be killing and digestion. So in here, again, you have the phagosome in here already, and the green dots will be the lysosomes. The phagosome will then fuse with a lysosome to form a phagolysosome. So that is when this green stuff attaches to this. Okay, and do note that lysosomes contain digestive enzymes which can destroy the pathogen. This will lower the pH, making the environment inside the phagolysosome very acidic. This is an effective way of killing or neutralizing whatever is inside the phagolysosome so it cannot infect the cell anymore. And that one will look like this. This is where the lysosome contents are released. And this one again, people, is because of the lowered pH caused by the enzymes. Do note that some viruses actually exploit the lowered pH to escape the phagolysosome and start replicating inside the cell. An example of this is influenza virus. Okay, so for influenza virus, they wait for that pH to drop so that they can go out of the phagolysosome and go to the cytoplasm. Once they reach the cytoplasm, they replicate themselves in order to infect the host cell. And lastly, phagocytes die. For phagocytes, we have to remember that there are two subtypes, right? We have neutrophil and we have macrophages. For neutrophils, they are the ones that die right away. And when we say die, it means that they went or underwent the process called as apoptosis, programmed cell death. They need to be removed because the granule contents inside their cytoplasm and some metabolites are harmful to the surrounding tissues. Thus, the process used for that is again apoptosis. Then later on, they are phagocytosed by macrophages itself. Okay, so they are eaten by their own kind technically. Macrophages, on the other hand, as we knew from the previous videos, have a longer lifespan and they withstand this ptosis from happening because they block the apoptotic pathway and they activate several survival paths so that they will not self-destruct. And that is why macrophages live longer than neutrophils. And that ends the whole process of phagocytosis. That's it for this video. I'll be seeing you again in the second part of the form. Bye!